I think of marine microbes as chemists. So there are the recyclers of carbon, nitrogen, oxygen. So lahat ng chemical component ng ocean dumadaan sa microbes. So microbes are really like this base of all life in the ocean. Kaya importante siyang makita at malaman kung gaano sila ka-diverse.
Magandang araw mga kapwa siyentista. Welcome sa BTS or ang Behind the Science podcast, kung saan kakausapin natin ang mga siyentista para ibahagi hindi lang ang mga resulta ng kanilang pag-aaral, kundi pati na rin ang kanilang mga behind the scenes ng kanilang mga pinag-aralan. Ito ay magsisimula sa kung paano nila naisipan gawin yung mga pag-aaral nila hanggang sa pag-publish nito. So this podcast is in partnership with UP Resilience Institute, and for today, ang ating guest ay isang biochemist, isang oceanographer, and Nat Geo explorer based in Honolulu with roots in Negros Island and Quezon City, Philippines. So recently, na-defend niya na ang kanyang Ph.D. in oceanography at the University of Hawaii at Manoa, and currently, his work is linked to molecular level interactions to ocean scale processes, focusing on marine viruses, bacteria, and plankton. So, without any further ado, let's all welcome Dr. Andrian Gajigan. Ayan. Welcome to the podcast, Kuya Adi.
Maraming salamat, Keith. Thanks for having me. I'm always listening to your podcast.
Wow.
During my free time, I'm listening to the BTS. So good job sa lahat.
Thank you very much sa
bumubuo
support,
ng
Kuya
BTS.
Adi. It really means a lot kasi syempre, we would want to share our science, 'di ba, to a greater audience.
True.
Ayan. So, siguro punta na tayo dun sa first muna natin. Alamin muna natin yung background mo. Let's go to the young and curious you.
Yeah.
So let's revisit your younger self. Kailan mo na-realize na gusto mong mag-work sa science sector?
Maybe after college na talaga. But starting like elementary and high school, I'm drawn into science subject. That is where I excel usually. But after my bachelor's, nag-try pa ako mag-apply for industry jobs. Although I find it difficult and also sobrang baba ng sweldo ng industry level chemist back at the time. Siguro nung master's na ako, saka ako na-realize na there's a career for scientists doing research na you can earn a living out of it. But the interest really starts from likely elementary and high school.
So, paano ka nag-arrive sa... Kasi, syempre, ang background mo, biochemistry eh. So, paano ka nag-arrive na napunta ka sa dagat?
Yeah, that's a great question. During my internship in undergrad, I visited Villanoy Marine Lab. I was an intern for this salt farm company. They're trying to understand yung microbes then on hypersaline lakes. So the way they do salt farming, diba? They just evaporate yung salt water, and they wanted to study yung organisms living on the salt lake in Bolinao. So we're looking for a space now we can do that, and that's where I found the Bolinao Marine Lab ng UP Marine Science Institute. And that's when I was like, had this moment na, oh my God, I want to work here. I saw people like coming from the boat, scuba diving. Like, I want to do what they do. Like, after field work, they look at the microscope or something. So that's really when I like got interested in marine science. Na, may sa field pala outside the lab na I can do both science and like environmental monitoring and field work.
So, medyo ano din kasi, medyo specialized yung field mo, Kuya Adi. So, hindi ka lang basta oceanographer. You're focusing more on the microbes. 'Di ba?
Yes. Yes, which is also a recent interest, I would say, because I worked with sponges and corals before. Um, Parang kung rule ko naman sa sarili ko, anything that has to do with marine science, any organism, kung ano mang organism yan, I will study it. It just so happens na from master's to PhD, I was so entailed with marine microbes.
So, ayun nga, so marine microbes. So for the information of our listeners, ayun, medyo nag-abot kami ni Kuya Adi sa Marine Science Institute, and I really looked up to Kuya Adi when I was at, at MSI. Kasi ayun nga, very interesting kasi din yung field na napuntahan ni Kuya Adi. With that, let's go now to the paper that we would like to discuss and share to our audience, 'di ba? So, in our segment called Kwento Mo Yan we will be discussing your paper entitled Diversity and Community Structure of Marine Microbes around Benham Rise Underwater Plateau in northeastern Philippines. So, Kuya Adi, in one sentence, can you explain your paper?
Yes, I've been listening to your podcast, and I think this is the hardest question that you're like give to your guests. I would say people wouldn't realize that ocean is full of microbes, like millions to billions of microbes, like small organisms that cannot be seen by the naked eye. So what we did is just look at kung ano-ano yung mga present na microbes sa region ng Benham Rise. At nakita namin na maraming microbes nga doon at may iba't ibang microbes depende kung gaano kalalim ka nag-sample ng water column. So it's also a factor of ano man yung chemical environment kung saan sila nakatira. So basically, that's the paper. And this paper is very special to me kasi this is, although this is like 2018 pa na-publish, so 6 years ago, this was my first research expedition. So ito yung, I think, nag-push me to also like do more research expedition after. So yeah, I'm thankful for the opportunity ng Benham Rise expedition that time.
Ayun, so, speaking of, ayun nga, expedition, di ba? So, bakit or paano ninyo na-formulate yung research na to in conjunction to the research cruises?
Yes. I don't know if may na-interview na sa podcast na to regarding Benham Rise. Meron na ba?
Parang
Meron na?
wala pa. Wala pa.
Oo. I think we could start from there because what happened is the Philippines filed an extension of our exclusive economic zone. Kasi parang yung exclusive economic zone natin, 200 nautical miles lang, di ba? Kalahati lang yun ng Benham Rise. But Philippines filed a petition sa UN to extend the continental shelf. So it covers now, noong 2014, 2012 ata, nanalo yung petition na yon. So buong Benham Rise na exclusively tayo yung may sovereign rights. So during that time, there was a clamor to study the resources that we have, what we have in Benham Rise. So that was 2014. So you know how like science takes so much time, diba? So the expedition happened 2014, pero the paper is published 2018. So a lot of people are studying yung mga resources nga, the corals, the fish. And on our part, we know importanteng component ng marine community is the marine microbes. So that's when we suggested, we should also look for the diversity of marine microbes in the region for us to have the complete picture from larger organisms up to the smaller organisms. Kung ano mang meron sa Benham Rise. That's how we were like, how the project started talaga.
So, bakit kailangan nating aralin or bakit important tignan din yung microorganisms present in the area aside from your corals and your fishes?
Yeah. The way I explain it to people, just because I'm also chemist by training, like Keith, I think of marine microbes as chemists. So there are the recyclers of carbon, nitrogen, oxygen. So lahat ng chemical component ng ocean dumadaan sa microbes. So microbes are really like this base of all life in the ocean. Kaya importante siyang makita at malaman kung gaano sila ka-diverse.
So, may effect din siya nun sa composition mismo ng nutrients and other uh, chemical compounds present dun sa oceans, di ba?
Yes, so it's a two-way system as well, 'di ba? So yung environment affects the microbes and the microbes also affects the environmental condition. So ganun yung effect nilang dalawa.
Nabanggit mo kanina yun nga, 'di ba? Nadagdag sa ating sovereign land or yung nasasakupan natin ang Benham Rise. So, bakit siya, for context, bakit important or ang laking bagay na nadagdag siya dun sa nasasakupan ng Pilipinas basically?
Yes. The way EEZ works, the exclusive economic zones, that would mean the Filipinos has the sole right to use all the resources in the region. So if we use the resources properly, kung ano man yung resources sa Benham Rise, na tayo lang yung makaka-exploit and makaka-utilize, that would help the Filipino people. But of course, that's another story if we can really tap whatever is in Benham Rise. But if we expand yung waters natin for exploration and we find something that of use, that would help the country.
Okay, so yun nga, very, kumbaga, mas marami kasi tayong pwedeng pagkunan, to summarize din yung sinabi mo, pagkunan ng
Yes,
resources.
exactly. Yes,
Oo.
exactly. Yeah. That's the simplified version.
Dito sa paper mo, Kuya Adi, did you secure or meron kayong funding? Kasi alam naman natin, hirap kumuha ng funding.
Yes. This project is actually not that earth-shattering. It's a simple project that we're really just like interested in doing. We had the small fund coming from the institute. The way funding works in UP Marine Science Institute, each lab has a in-house funding. So the institute coming from its fund gives some amount of money um, to each lab. So that's what we use for sequencing. Um, Usually yung gamit namin is we already have because yung lab naman marami naman siyang ibang projects. So we already have the equipments, the supplies. The most expensive part lang during that time was the sequencing part. Sequencing of the genetic material of the microbes so that we know what's in the water.
Can you enlighten us, Kuya Adi? Parang, paano mo or paano nyo kinuha basically yung microbes? Kasi syempre, maliit siya. Hindi mo nakikita with your naked eye. So, paano nyo siya kinolek?
Yes, that's a great question as well. Our best friend as microbial oceanographers are filters. So we have sets of filters with different pore size. So they have different like size ng butas in Tagalog. So what happened is we use a filter with a very, very, very small pore size so it can trap the microbial cells. So we just put the seawater on a
So,
filter and
in...
everything that each cells will be
Oh,
trapped
okay.
on that filter. Kasi it's bigger than the size of the holes in the filter. So basically that's the sampling part.
Okay. So, ano siya? Tinotroll nyo ba siya or parang hinihila sa bangka? Or nagko-collect lang kayo ng tubig at certain depths?
Yeah, so similar lang din sa typical sampling ng oceanographers, di ba? So ni-skill bottle lang, sampling seawater at different depths using a bottle. And then once it's on board, we can collect the seawater na and filter on board. We can do also trolling of or deploying an instrument that can filter seawater in the ocean. Pero that time, we're not that advanced yet. Pero there are machines that you can deploy and it can filter seawater on site. Pero for this project, we just get water on board and then we filter it along. Yes.
So, dinala nyo pa siya from the boat itself going to MSI, yung tubig, or you filtered on site and then just stored the filter?
Yes. So the latter. So we filtered it on board the ship and then we flash frozen. So nagdala kami ng liquid nitrogen on the ship. So we want to
Ah.
freeze whatever diversity in there because if you allow it to be there alive, may magko-compete na microbe, may mga magiging successful, mag-iiba yung community. So we need to freeze whatever we found. So we soak them in liquid nitrogen.
In terms of, nabanggit mo na yung diversity. So, how diverse are the microbes in Benham Rise?
It seems na simple yung question na ganyan, pero really it's really a difficult and
Oo.
challenging question to answer. Kasi it all depends on the method that we use kung how resolved our method can detect the diversity of microbes in the region. So I cannot tell for sure. And also, different labs use different methods, so it's hard to compare. Um, Another palang hindi ko nabanggit earlier, another reason why we came up with this project is there's have been global sampling around the world ng marine microbes and plankton, bacteria, viruses na hindi kasama yung Pilipinas. So it's quote unquote global sampling, pero they did not include Philippines, which is the center of marine biodiversity, diba? So we're curious whether
Yes.
the marine microbes is also more diverse here than around the world. But again, different methods yung ginamit nila. Kami dun, slightly different. But that's one of the reason why we did it. Yeah, so I don't have a clear answer to your question, but hopefully in the future we can really have this standardized method to compare across the board kung saan talaga diverse. I would say we don't
Oh.
know.
Pero ano, I mean, syempre, nabanggit mo you're comparing at different depths. So,
Yes.
within, yun nga, comparing across depths na same site, I mean, may difference ba yung microbe community
Yes.
within those depths na tinignan ninyo? And,
Yes. Yes. So meron. So merong difference in community across depth and usually mas diverse at mid depth. The reason why, or at least the hypothesis is, at mid depth, marami ng niche, yung tinatawag na ecological niche, which is marami lang pwedeng pagbahayan yung microbe. Marami lang different environments at mid-depth. Kasi dito mo na makikita yung mga marine snow. Dito na iba-iba yung lahat na nagpo-fall down from the surface water to the mid-depth. Dumadami sila. So marami na nga yung iba't ibang ecological niche yung microbes. So mas diverse sila at mid-depth. I hope I explained that.
okay. So, I really
Yeah.
thought, oo. Akala ko mas diverse near the surface. Yung pala hindi.
Hindi. At least for bacteria and archaea, it's mid-depth.
And how deep is mid-depth for context sa ating mga listeners?
The yung sampling namin until 300 meters lang. But the global sampling na sinabi ko before, meron silang mga sample deeper, at a deeper level. Pero the same conclusion pa din. Mid-depth usually yung highest diversity. Yeah, pero malalim na rin ang 300 meters if you want to think about it, diba? Kasi sobrang lalim na rin ang 300 meters.
Isang ano na yun, isang high-story building na yun.
Yeah.
'Di ba? Parang, oo. So, ayun nga. So, nabanggit mo na very special yung paper na ito. So, let's talk about the challenges na na-experience mo while doing this paper. Ano yung mga memorable na experiences mo dito sa project na to?
Yes. Actually, yung spot pa lang to join the cruise was challenging na. Kasi we only have limited spot for people to join the cruise. And I was not even part of oceanography back then. I was in a different lab. So we suggested this to the chief scientists and the other professors. And tinanggap naman nila. So that was first. And I'm really glad na naisama nga itong proposal namin to look at the microbes. The second challenge was the actual filtering itself. If you go down to the water column, nabanggit ko nga, di ba, marami ng marine snow. And yung pore size ng filter namin are very small. So it will clog the filter very fast. So for us to get our biomass enough, we need to filter liters of water na it will take so much time to filter. So, naalala ko nung time na yun na nagko-cruise, natutulog lang ako dun sa lab kasi I need to kailangan ko bantayan yung setup ko every time. And then when we reach the next station, that's when palang matatapos yung previous sampling ko. So, you know how oceanographic research expedition works, di ba? Medyo gipit sa oras and you wanna maximize yung sampling kasi nga naman sobrang mahal mag-research expedition. So, definitely kulang ako sa tulog back then. And it was my first time to also do this kind of sampling. So everything was first. My first research expedition, first venture into marine microbes. Yeah, so everything was first. And I was like, happy na it worked. Yun.
Ayun nga, and ang dami din kasing ang daming papers na lumabas na connected dito sa research expedition na to, if I'm not mistaken. Tama, di ba?
Yes. Medyo, I think may 3 or 4 pang paper na lumabas on benthic component, on fish, on diversity. Yes. Iba't iba yung papers na lumabas regarding this expedition. And this was the time back then na wala pa tayong ships. Wala pa tayong fleet. Wala pa tayong research fleet. So, ang ginagamit pa lang namin is yung BFAR ship. So, ngayon, maswerte na yung mga marine scientist students kasi we have different ships that we can use. You can even use the GT Velasquez, the BFAR ship, and yung in-house ng MSI. Pero back then, yung DA BFAR lang yung meron.
Oo nga, naalala ko to, yung sobrang hindi naman poor, parang resource challenged lang tayo ng mga time na yun.
Yeah, especially if you join cruises internationally, you will realize how challenged tayo in terms of resources and space. But to make do with what we have. So yun naman yung ating panlaban.
Yun nga. So if it's anything, Filipino scientists are they make use of resources very well. Yes, resourceful.
Aha.
Kasi,
and they're very
yun na yung pangyabang natin.
Aha. Because we're used to having in a bad, non-conducive situation, we know how to make do with little. Yeah. I would say. Not that we want to romanticize that kind of working situation, diba? We wanted things to change as well.
Ayun. So, since nag-share ka na rin naman ng experiences mo, so ito, the good, the bad, and the ugly. 3 memorable experiences while doing this research under those 3 categories. Start tayo sa good, Kuya Adi.
Good, I was happy that I was mentioning earlier, this was my first research expedition. I'm happy na we were able to publish the result of this expedition. So those are the good things. Yung bad, I think, just hindsight, no? Yung working situation, sa abri na lang yung working situation. Sa bad, I think yung kulang ako sa tulog during the whole research cruise. But that's how things work pag research expedition naman. Lahat tayo talagang pagod. Kasi nga, we're trying to maximize the time that we have in the open ocean. So the ugly part, which is recent ko lang din na-realize, is yung ating working condition on board, di ba? Na sana ma-improve natin. Limited yung space natin, the setup, the labs, the capacity of what can do. I'm really happy na the national government and UP is now like trying to push yung national research fleet. So we're now investing into large ships na we can use for expeditions like this. Pero naalala ko, medyo TMI, pero kasi yung very particular ako sa banyo, sa room. Grabe yung banyo talaga ng DAB FAR. If you're really catering to like large amount of people, di ba? So I think medyo mahirap siyang, I'm sure for the logistics ng BFAR on how they can manage mga how many people in one boat sharing the same restroom and toilet, di ba? So yun yung, challenge.
So, ilang ano ba? Ilang CR ba meron sa ano? Sa
The thing though is,
Velasquez?
sa Velasquez, I don't know, mas malaki yun eh. Kasi ano di ba yun, galing sa scripts sa UC San Diego. Yung
Ay, oo,
DAB4,
oo.
di ba? I don't know if you've been to the
Oo
DAB4
nga.
ship. Different siya. So, shared yung bathroom ng men, shared yung bathroom ng female. Tapos siguro mga dalawang toilet or tatlong toilet, apat na shower for not just the scientists but also all the crews ng research, ng vessel.
Ah.
So pag naglag yung isang toilet, yan, yung mahirap. But it's just one of those things na kaya natin yan. And also, because of the size of the ship, we can only bring limited amount of water. So, kailangan ration din yung paliligo, paghuhugas. Kasi nga, limited yung water na pwedeng dalhin in the open ocean. So, those challenges ba niya parang very third world, but still, hopefully, and I'm happy again to say na we're going in the right direction, investing in more and high-end ships and vessels.
So, for context, gaano katagal yung mga ganitong research expeditions? Usually.
It all depends
para lang
on
magka-idea sila
objectives.
na.
Yeah. Pag ganito, nung kami mga weeks mga 2 weeks ata, kaming nasa open ocean. Depende talaga sa boat, gaano kalaki yung boat, what are the objectives of the expedition. Kasi may mga expedition na more than 30 days na umaabot ng 45 to 60 days. Imagine you're on the ship 45 to 60 days. But yeah, weeks to months, I would say.
Weeks to months. So, pero ano yun, kasama na dun yung pagpunta mismo dun sa ano no? Sa site.
But yes.
Yun, for context lang ulit, gaano kalayo si Benham Rise in terms of days from mainland Luzon?
Yeah. You know what? I forgot ngayon. Pero siguro, I would say, pero hindi
Based on your
mainland Luzon din
experience?
lumabas eh.
Oo.
Ko 2 days, 3 days. Kasi galing kaming Manila Bay eh. Galing kaming Navotas Port. So, ikot pa kami.
Umikot pa kayo. Oo.
Oo, hindi kami galing sa Aurora side. Yeah, 3 days. Siguro kung galing ko sa Aurora na port, it would just take a day. Pero kung galing kang Manila Bay, it will take 2 to 3 days maybe.
Oh, okay. Ayan, so ganyan kalayo ang tinravel just to collect samples. I mean, ayun nga, it's part of our job as scientists and ayun nga, gaano man kalayo.
Yes. And also, there's a lot of perks as well. I mean, not everyone could experience yung feeling of like not seeing the city and congested sunset and sunrise. I think every time nagigising ka, the sunrise is there. Maganda din yung sunset sa gabi. And Dahil wala din kaming signal, so wala kaming phone, we're not distracted in any way by social media.
Oo.
It's really a detox from the city, from social media. So I think that's a nice perk of it, I would say.
Okay. So, yung balik tayo sa paper, Kuya Adi. Yung
Yes.
samples ninyo, so nabanggit mo kasi kanina yung pagpapa-analyze, yung ano
Sequencing.
tawag dito? Sequencing. Oo.
Mhm.
So, gaano siya katagal or did you outsource it or meron tayo sa UP?
Nung time namin, we outsourced yung sequencing. So we extracted the DNA from the microbes and then we sent it away in Hong Kong for sequencing. Pero that's the time also when the Philippine Genome Center is starting. So meron na silang mga machines and meron na silang mga staff, pero hindi pa ganon ka-establish yung sequencing facility ng Philippine Genome Center. But I believe now, Philippine Genome Center is well established, so they can do this simple kind of sequencing. But before, we said it in Hong Kong or other places. It's also faster. Kasi pag nag-send kami dati sa PGC, it will take more time.
So,
Yeah.
what type of sequencing yung kasi background parang alam ko parang merong iba't ibang types ng sequencing, 'di ba?
Yes. For this particular project, what we did, we collected the DNA, not the RNA, so the genome ng microbes, so DNA, and we sequenced a region, a specific region lang. I think, if I'm not mistaken, short region lang siya, mga 250, 300 base pair ng DNA. It will allow us lang to identify what's in there kasi we can compare the genome sequence to a database. And that database will tell us what organism is in there. It's a matter of collecting all those sequences from different microbes and then comparing those sequences to a database, and it will tell you what type of microbes are in there. So, yeah. And it's only a portion of a genome. Sobrang haba ng genome. Like millions, sometimes billions. And we're only like sequencing a small portion of it, enough for us to identify what type of microbes are in there.
Oh. Okay, ang dami nun. I can only
Yeah.
imagine the amount of data that you have.
That's, I think, also the challenge, yung bioinformatics part on how you analyze the sequence. That's another skill to learn as well. And a lot of scientists now are like learning how to code. Pero yung bioinformatics specific for like biology studying genome and other sequencing data. But yeah, everything is out there. You can learn it by yourself. But yeah, yung learning curve lang medyo steep if you're like trying to learn it first.
So requirement na ba na marunong ka mag-code if you want to go to bioinformatics? Gano'n na ba siya ka-competitive?
So I think every field in science, at least people who wanted to like any career in science, have a career in science, need how to code. Biology man yan, ecology, basic stats needs to be coded. Also, yung paggawa ng mga figures, graphs, it will be easier if it's automated. So the way you automate that is by coding. So yon.
Okay. The future is data science talaga, no?
That's another thing to unpack as well. And also, we'll take as an hour to like discuss that as well. Pero it's a skill na people shouldn't avoid, yung coding. It will help everyone, scientists in particular. Yeah.
Okay. So, at what point did you realize na yung results for this particular paper is sufficient enough na to publish?
Yeah, I think on our part, very straightforward lang kasi yung objective namin eh. So like sample water, look at the diversity, and when we did that, it's ready to publish. Of course, hindi siya tulad ng ibang studies na mas hinahanap yung mechanistic. Mas more on the mechanism of certain scientific phenomena, which is a much higher level of science, diba? This paper is discovery-based.
Mm-mm.
Um, so when we know na yung objective namin is to just discover and to like describe what's in there, then we're ready to publish. And I think this needs to be space for that type of science. Not every science needs to be trying to understand the mechanism, 'di ba? Pero
Mm-mm.
yun talaga yung mas kailangan natin, 'di ba? The
Yes.
how
Mm-mm.
things work, 'di ba? Mas mataas talaga yung level ng science nung if you answer how things work.
Mm-mm.
But still, because we're just starting to just go to this site now never have been studied,
Mm-mm.
okay lang yung mga discovery based just to give you a baseline of
Mm-mm.
what's in there. Yeah.
Ayun nga, ayun. Nabanggit mo na, important yung what if you're new to a certain place kasi di mo alam
Yes.
kung anong meron eh. So,
Yeah.
you need to start at the very,
Excellent.
very di ba?
Excellent analogy. Bago ka kapag how, you need to answer what.
Mm-mm.
Diba?
Ayan, Kuya Adi, I know that you have just finished your PhD and you're moving forward in the field of microbial oceanography. If we can coin that. So, what are your further plans moving forward in conjunction to this research?
Yeah. At least for this part, I wanted to dive into yung naging research ko kasi ng PhD, ano na, marine viruses.
Mm-mm.
Aside from bacteria and archaea, marine viruses na yung study ko nung PhD. And I wanted to look now for my postdoc, the larger marine disease ecology. Kasi a
Mm-mm.
lot of marine organisms are also getting infected because of different types of pathogen.
Mm-mm.
That's a field that I wanted to like dive into for the next steps of my career. It will help kasi maraming diseases ang unknown pa yung cause that impacts different types of livelihood, coastal communities. So it's a great niche to study. Yun. In terms of
Ayun.
microbes, we need to study more microbes around the Philippine archipelago because we have a lot of diverse environmental and oceanographic conditions. We have yung, I know a lot of people are studying microbes in carbon dioxide seeps in
Mm-mm.
Batangas, 'di
Mm-mm.
ba? Meron tayong mga anoxic regions. So
Mm-mm.
different environments will give you different microbes. So it's interesting to look at different microbes and what they're doing in different environments. But on that aspect, Philippines has like inexhaustible interesting environments to look at.
Tsaka ayun nga, ang dami kasi sobrang varied din. Extremes ang environment natin dito sa Pilipinas.
Yeah. And we're not yet talking about mga vent system na hindi pa
Mm-mm.
natin nakikita, hydrothermal vents na malalim na don't have a capacity to look at.
Mm-mm.
Also, the Philippine Trench, na the
Mm-mm.
third deepest trench. Diba? So, yeah, there's a lot of things that needs to be done.
Okay. So, before we let you go, Kuya Adi, what would be your advice to aspiring researchers that would want to explore this field?
Yeah. Um, Sige, yung practical advice ko earlier is like learn coding, di ba? I
Mm-mm.
think that's a practical advice. Pero nung pinag-iisipan ko tong question na to, what comes to mind is like there's no shortcut. Laging kailangan talagang hard work in
Mm-mm.
terms of your master's or PhD. Wala talagang shortcut. Kailangan mong pagdaanan lahat ng paghihirap before you
Mm-mm.
get to a point na you have this learned wisdom um, to like push through and like push the boundaries of science. So I think yun yung remind ko din sa sarili ko kasi a lot of times I'm impatient. So sabi ko, there's no shortcut. I need to like um, go slowly. Kailangan ko pagdaanan and like tutukan yung mga each part of this whole path, if that makes sense. So I think for all aspiring um, scientists out there na trying to just start their career, for sure at the very beginning, everything that you're just starting, it's hard at the beginning. Wala namang madali sa simula. So
Mm-mm.
after a while, it will be worth it. And it's just a matter of gaining confidence. Pag nakakuha ka na ng confidence, finishing a particular task, you'll be more bold in like doing more like larger, bigger scientific questions after. So yeah, I think that's my
Ayun.
main advice to our listeners.
Ayan. Thank you, Kuya Adi. So, do you have anything to promote or to advertise para sa ating listeners?
um, One thing also when I was like thinking about this question is like midterm election is coming up, diba?
Mm.
So, do politics is affecting our science? We're talking about yung working condition ng scientists. So, ang ipopromote ko lang is vote wisely this midterm election and they should look at um, the Makabayan block because they're promoting really mga they wanted to create laws for like to help yung ating mga health workers, mga scientists, workers, so everyone. So they should look into it. Yun. Yung aking promote. Because it's not, the politics, our everyday life affects our science, I
Mm-mm.
would say.
Thank you very much, Kuya Adi. Sobrang dami kong natutunan. And we wish you well in your future endeavors. And sana makabalik ka dito sa podcast for another discussion on another
For
paper.
sure. For sure. I would love to do it again. Thanks so much, Chris.
Okay, sige. Thank you, everyone.